My name is Chelsea Rochman. I'm a professor at the University of Toronto. I'm a researcher, so I study marine ecology, and being here in Canada, I study a lot of freshwater ecology these days. I had the opportunity to go to the Great Pacific Garbage Patch in 2009 in the summer on the New Horizon with the Scripps Institution of Oceanography. At the time, I was a first-year graduate student actually at the university down the street, San Diego State. And my role was to help understand the chemicals on plastics and how they may have impacted organisms. So I was sampling animals to look for chemical additives in them, as well as helping others on the ship quantify the plastic that we saw on board. How would you describe a 24-hour day for you while working on the New Horizon in the Pacific Gyre? A 24-hour day did not involve a lot of sleep, and it often did not involve sleep like at one time in an 8-hour sequence. So I was on day crew, so I was lucky enough to not be working throughout the night. So I would tend to go to bed at a normal hour. So let's say I wake up in the morning and I eat breakfast in the galley of the boat. And then I would go outside and I would sample. I was helping Miriam actually sample surface water in the nets. I was helping Pete sample fish with the Ozeki trawl. I would spend a lot of my day going outside, helping with the net, and then coming inside to help prep the sample, write things into my computer to note where the samples were. I was crazy about labeling, so I was like the one that would run around and relabel everyone's samples. We would have lunch at a normal time. Food was great, so we would eat our normal meals. And when I had downtime, I would read or study or watch TV. But the reason I never slept is because the coolest stuff happens at night. So I told everyone to wake me up the minute you see anything really cool. So the coolest thing that happened one night was bioluminescence as far as the eye could see. The sea was glowing with every little wave, and a clear sky of stars. You couldn't even tell the difference between the ocean and the sky because it all blended in this twinkling bioluminescence. And so it was that type of stuff that I would wake up for at night and then lose sleep because I was having so much fun. When I was out there, the animal that I was looking for my research was not charismatic, and it was something called a SELP, and it is the jelliest, gooeyest, weirdest looking thing. It's actually it's a zooplankton. It floats around in the currents, and they tend to accumulate onto each other, as you can actually see in this picture. You pull them up in the net, and you have to be very careful because they'll just splat into boogery goo if you're not careful. So we would try to collect them with hand nets so that we could have one at a time. But yeah, I literally brought home a bunch of little loogies in tinfoil to then analyze the chemicals later. But they're also really important animals, and because of how they feed, they actually are vulnerable to eating microplastics. So that's why I wanted them. But super cool organisms out in the middle of the ocean. Also bright blue animals that blend in with the sea. This is really neat. One of the reasons I got interested in studying plastic in the ocean is because I was really interested in their toxicity. I care about fish, I care about wildlife. I wanted to know how it was impacting them. Some of the things I study are how chemicals associated with plastics impact the animals. So plastics are made of lots of different chemicals, and then when they enter the environment, they're like magnets for the pesticides and flame retardants and metals and other chemicals in the water. I wanted to understand how those chemicals leach from the plastic, so simply come off the plastic and go into the water, go into the animals. I also want to understand how the chemicals from plastic move through the animals. So when a plastic is eaten by an animal, the plastic may be excreted, like when it goes to the bathroom, for lack of a better term, but the chemicals might stay behind in their tissues. If the chemicals stay behind, and let's say that fish is now eaten by a predator, the chemicals transfer up the food chain to the predator. And that predator is more hungry than its prey because it's larger, so it might need to eat 5 fish. Now it has even a higher amount of chemicals from the plastic than the original fish because it ate 5 fish that ate plastic. So we study how the chemicals leach into the animals and whether they biomagnify, meaning magnify or increase up the food chain. Now, who eats at the top of the food chain? We do. So that's another reason why it's important, because when we eat tuna or other types of fish from the sea, we can also be impacted by the plastic that we let go in the ocean. Some of the biggest challenges when we're at sea is sampling the sample types you want to sample. We are limited by our technology, right? If we want to sample water, we often attach a rope to something on the ship and send it into the water and drag it behind the boat. If we had autonomous things like water samplers to be able to take samples, not just at the surface, but further down to understand how the fish that don't feed from the surface might be impacted by plastic. Also, humans can't go as deep as machines can. And so being able to send something down deeper to get samples is also really important.